Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on evidences for semi-conservative mode of DNA replication. The evidences for semi-conservative mode of DNA replication was given by three scientists. They are Messelson and Stahl and also Keynes. So let's see the first Messelson and Stahl's experiment. Okay. So Matthew Messelson and Franklin Stahl, they worked on E. coli bacterium and they proved that the DNA replication is a semi-conservative mode of replication. Okay. So semi-conservative mode of replication is nothing but the newly synthesized DNA strands, they are consisting one parental strand and one newly synthesized strand. So that is nothing but semi-conservative mode of DNA replication. So that is proved by these two scientists. Let's see the experiment. Okay. So they worked on E. coli bacterium. So they worked on E. coli bacteria. They used a technique called density gradient centrifugation. Density gradient centrifugation. So they grown the E. coli bacteria in a medium containing nitrogen isotopes. They have used two isotopes. They are nothing but N15 and N14 isotopes. So for initially they grown the bacteria in a culture media, the media containing N15 isotopes. Okay. So now all the DNA present in an E. coli that get converted into heavy DNA due to the presence of N15 isotope. Okay. So now the bacteria isolated and they are transferred to the another medium, that media containing N14 nitrogen. Okay, that media containing N14 nitrogen. Now again the replication cycle occurs. So after first replication cycle, he got hybrid DNA. After first replication cycle, he got hybrid DNA. Hybrid nothing but, so it is having one heavy nitrogen strand and one lighter nitrogen strand. Okay. Lighter DNA is nothing but which is having N14 nitrogen. So that's why the density of this strand is lighter than the N15 DNA fragment. Okay. So they got all the DNA is having hybrid DNA after first replication cycle. Then they were isolated and they were grown in the media containing N14 again. Now the third replication cycle occurs. So here we are going to get one hybrid DNA strand and one lighter DNA strand. Hybrid DNA is nothing but here it got one higher density strand and one lower density strand. Okay, one lower density strand. Okay, so that's why in the third generation they get one hybrid strand and one lighter strand. So this experiment proves that DNA replication is a type of semi-conservative mode of replication. Semi-conservative is nothing but a newly synthesized daughter strands. They are consisting one parental strand and one newly synthesized strand. That is nothing but semi-conservative type of replication. Okay, so let's see the second experiment, that is Keynes experiment. So that is Keynes experiment. Okay, so John Keynes, he also worked on E. coli bacteria. He used the autoradiography technique to visualize the E. coli bacteria. He also proved that the DNA replication in E. coli is semi-conservative type. Okay, so he also proved that the DNA replication is semi-conservative type. Okay, He used the radioactive thymine. So let's see the experiment. So John Keynes, he also worked on E. coli. He grown E. coli bacteria in a medium containing radioactive thymidine bases. Okay, so he grown the E. coli bacteria in a medium containing radioactive thymidine bases. So all the DNA present in the E. coli, they get converted to radioactively labeled DNA due to the presence of thymidine bases. Okay, all the DNA present in the E. coli bacterium, they get converted to radioactively labeled DNA due to the presence of thymidine bases. So then he lysed the bacterial cells. He spread the DNA present in the E. coli onto a slide. So the slide was covered with a photographic film for autoradiography. The slide was covered with a photoradiography film for autoradiography technique. So he kept the slide in a dark place. Okay, so after developing the film, he found the replication bubble in DNA. So E. coli having the circular DNA, so he found the replication bubble under the microscope. So here you can see the origin point. So this is the replication bubble. So it moves in two directions. Okay. So in this direction and this direction. Okay. It moves. Replication bubble is moving in both the directions. He found that. Okay. So he also observed that the newly synthesized strands, they are having radioactively labeled thymine bases. So you can see the daughter strands. So you can see the daughter strands. So one strand among the daughter DNA is radioactively labeled DNA. So here also you can see the outer side DNA molecule is a radioactively labeled DNA. Here inner side DNA is a radioactively labeled DNA. So this proves that the DNA replication is a semi-conservative type. So, and also proves that the DNA replication in Escherichia coli is bidirectional. It is also a bidirectional type. Okay, students. So, these are the experiments have made by these three scientists. They proved that the DNA replication is semi-conservative type. Okay, so if you like this video, please share this video with your friends. If you not yet subscribed my channel, please subscribe. Thank you so much.